Hi everybody, my name is Rodrigo Ochoa, and today I'm going to present the work Virtual Screening of Potential Kinase Inhibitors within this particular signaling pathway of Leishmania. So this project is a collaborative work between Colombia, the University of Antioquia, and Argentina with the University of Buenos Aires. So the main motivation of this work are the neglected tropical diseases. So they affect millions of people around the world. And there have been multiple alternatives to reduce the global burden of the disease, most of them related with public health policies as a preventive strategy. But unfortunately, we need more treatment alternatives, better drugs and vaccines to eradicate the causal agents like parasites. And one of these one of these diseases is Leishmaniasis. That is an NTD caused by different species of Leishmania. So this disease is present in around 99 countries around the world and has a very complex life cycle, and it's transmitted by an insect vector. So it causes different clinical forms, but one of them is the cutaneous Leishmaniasis, and the second one is the visceral Leishmaniasis, that is the second largest parasitic killer in the world. So there are many challenges around that. For example, the drugs available are very old, toxic, and expensive. So there are resistance of the parasite, mainly in endemic regions. The public health regulations are not sufficient to eradicate the disease, and requirement of hospitalization conditions is not generally available because also of the population affected by the diseases. And finally, there are not a vaccine available because of the complex life cycle of these parasites, and there is a lack of information about molecular targets. So regarding the targets, we are working with kinases and potential signaling pathways in the parasite that based on the expression of many of them, there are hints about the presence of apoptosis-like or PI3K-AKT pathways. And the search for homologs of the most relevant kinases definitely opens up the possibility of using them as potential molecular targets, and also because of the relevance they have in other diseases. So there are different computational strategies, and we have been working in an integral computational approach where we have focused part of the research for target discovery using comparative genomics. Then we have worked in target interactions using protein-protein interaction networks, and we have also applied structure-based methodologies based on modeling protein structure from different Leishmania species in order to look for potential inhibitors. And for virtual screening approaches, we have used three-dimensional information, in this case molecular docking, where based on the conformational pose of the ligand, we can infer a score and then predict a potential free energy of binding. So normally this is done for a big set of molecules in order to have a list of compounds and then proceed to experimental validation. So the goals of this work is implement an integrative bioinformatics strategy focused on a set of parasite kinases that can be associated to this pathway, the PI3K-AKT pathway, and try to do the correlation based on information of the mammalian counterparts. So the first part involved the comparative analysis, the research of potential homologs of some particular kinases. Then we apply modeling and molecular docking strategies using structural information. And finally, we focus on essentiality evaluation using network data. So in the general methodology, we start with a best reciprocal hits protocol in order to search for potential homologs. Then for the screening, we first create a virtual small molecule library around 45,000 molecules, and then we model the kinases structures and also select potential druggable pockets within the structure. Then all of them are subjected to molecular docking. Based on the results, we apply a clustering analysis and compare also some inhibitors with reported compounds, and then we finish with the study of kinases and essentiality in protein-protein interaction networks. So regarding the homology results, we applied the best reciprocal hits protocol, and we found orthologs of 5 particular kinases that are AKT, GSK3, AMPK, mTOR, and CDK. So we found homologs of them in 5 species: Mexicana, Major, Brasilensis, Infantum, and Donovani, based on the UniProt ID codes. And the presence of possible signaling pathways is also confirmed not only because of these 5 kinases, but also for additional homologs that we found within the root. Regarding the modeling, we model each Leishmania kinase, and we took into account quality scores like the Q-mean and also the druggability score of each predicted pocket. So this is an example of the models generated for Leishmania brasiliensis, and here you can see the metrics for some of the kinases. And the idea is that with the models that we obtain per each species, per each kinase, we subject that to virtual screening using a server called the tag drug discovery attack, where we can screen the library of compounds within the druggable pockets. So after that, we found the common molecules within the group of kinases, and we identified the main scaffolds that can be used as starting points for optimization phases. And also the compounds were compared with known kinase inhibitors using the LIQ server. So for that purpose, we applied the Tanimoto similarity criterion, and we selected 3 molecules that are good candidates based on the virtual screening. Finally, we checked the roles of these kinases within the protein-protein interaction networks, and we calculated that for 3 Leishmania species, Major, Infantum, and Brasiliensis, looking for highly central and essential targets. So based on topological metrics like the degree or the bottleneck, we identify that these kinases are quite important within the molecular pathways of the parasite, and based on also on the literature findings, they can be prioritized as molecular targets. So it is plausible that the parasite can trigger similar signaling pathways as survival mechanisms like apoptosis-like events, among others. So finally, it's important to summarize that kinases are molecular targets that can be very useful to look for new potential treatments. Actually, in our group in Colombia, we are focused right now in the AKT kinase and looking for allosteric sites to try to inhibit the enzyme. And also our protocol is reproducible and can be applied in the context of other pathogens. So I want to acknowledge both institutions, all the researchers involved, and the Cabana group also for providing the connection between the researchers. So thank you very much for your attention.